Hello students, welcome to EPG Pathshala. I am Dr. Sanyog Jain from Naipur Mohali. Today we are going to discuss about a module entitled Basics of Protein, Protein Architecture and Recent Scenario of Protein and Peptide as Therapeutics under paper NDDS Part 2. So these are the learning objectives. We are going to discuss introduction of the proteins and peptides, introduction and classification of amino acids which are the building blocks of the proteins, basics of protein and protein architecture, types of proteins and protein organization, different levels of the protein structure like primary structure, secondary structure, tertiary and quaternary structure, introduction to the protein pharmaceuticals, and lastly the classification of protein therapeutics, their examples and their clinical use. So let's start with the introduction. Protein is considered as a biomacromolecular polymeric chain with repetitive units of the amino acids linked via peptide bonds. So amino acids are the basic units of the polypeptide and proteins, and out of almost 300 naturally occurring amino acids, only 20 consists of the backbone of the proteins and peptides, and interestingly all these 20 genetically encoded amino acids which forms the proteins, they are L-alpha amino acids, and out of these only 19 are the primary amines, and only one, that is the proline, is the secondary amine. And further, all these 19 amino acids, they have the chiral center except the glycine which has the achiral center, and its natural configuration of alpha carbon is L. So proteins also occurs in great varieties, thousands of different kinds ranging in the size from relatively small peptides to huge polymer with molecular weights in the millions may be found in a single cell. So genetic information is inherited by DNA which is eventually transformed from generation to generation, and that has several proteins. So the examples include histones and non-histones. So histones are the very basic proteins which supports the coiling of the DNA, and these are subclassified as H1, H2A, H2B, H3 and H4. So all the proteins are constructed from the same ubiquitous sets of 20 amino acids covalently linked in a characteristic linear sequence. So as I told earlier, the amino acids are the building blocks for the protein. Each amino acid residue is joined to its neighborhood by a specific type of covalent bond that is known as peptide bond. Amino acids share some common structural features like they are having L-alpha configuration, and all amino acids have a one carboxylic acid group and one amino group, and they are bonded to the same carbon atom that is known as alpha carbon atom. They differ from each other in their side chains or R group. So R group can vary in structure, size and electric charge, and that ultimately influence the solubility of amino acids in water. Classification of the amino acids. There can be several ways to classify amino acids. First classification is based on the location of the amino group, means position of the amino group relative to the carboxylic acid group. So they can be subclassified as alpha amino acids, beta amino acids and gamma amino acids. Another way of classification is based on the properties of the side chain. So in that, the first can be the non-polar side chain, where the side chain is non-polar in nature. The example include glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, tryptophan, etc. The second classification can be the amino acids which having the polar but non-ionizable side chain. For example, the amino acids which contains hydroxyl group, sulfhydryl group, amino group. These groups are polar in nature but they are non-ionizable. The examples include serine, threonine, tyrosine, cysteine, asparagine, glutamine, etc. Third category, amino acids with polar ionizable groups. So these amino acids have the polar and ionizable groups. For example, either they can have the carboxylic acid group or they can have the amino group which are ionizable in nature. The example include aspartic acid and glutamic acid. These two acids have the ionizable carboxylic acid group, and lysine, histidine, arginine, these having the polar ionizable amino group. So this shows the basic property. However, the amino acids which have the acidic group, they shows the acidic property. Yet another way of classification of amino acid, it is based on their nature. So they can be subclassified as hydrophilic amino acids, hydrophobic amino acids, essential or non-essential amino acids. So first we discuss about the hydrophilic amino acids. So these are the amino acids which interact extensively with the solvent via ionic and hydrogen bonding and present on the peripheral part of the protein. So the hydrophilic amino acids are also present in the interior of the protein, which can be responsible for the formation of the salt bridges. The examples of hydrophilic amino acids includes aspartic acid, glutamic acid, lysine, arginine, histidine, asparagine and glutamine. Hydrophobic amino acids are mainly present in the core of the protein, like unlike the hydrophilic amino acid which preferably find onto the periphery of the protein, these hydrophobic amino acids are present in the core of the protein, or we can say they are buried within the folded protein. So some hydrophobic residues can also be or partially or entirely exposed onto the surface. So some exemptions are there, like methionine, isoleucine, leucine, valine, phenylalanine, tryptophan, tyrosine, alanine, cysteine and proline. Amino acids can also be classified as essential amino acids and non-essential amino acids. We all know that these essential amino acids are known as indispensable amino acids because they cannot be synthesized inside the body by the de novo pathway. So that's why they are need to be supplied in the diet. So based on this, amino acids forms the three major categories: essential amino acids, conditionally non-essential amino acids and non-essential amino acids. So the conditionally non-essential amino acids are need not to be obtained by diet under the normal conditions. They can be synthesized inside the body, but in certain special circumstances they need to be supplied by the diet also. This table shows the various examples of essential amino acids, conditionally non-essential amino acids and non-essential amino acids. So if we see these essential amino acids, these includes histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, etc. These conditionally non-essential amino acids include serine, proline, glycine, glutamate, arginine, asparagine, tyrosine, etc. And non-essential amino acids includes alanine, aspartate, glutamate and cysteine. Another interesting fact about amino acids is their deuterionic behavior. Amino acids contains both amino as well as carboxylic acid group. Therefore, they exhibit a dipolar ionic form where the two polar groups are ionized. Although amino acids are amphoteric in nature, but they can react either as an acid or base in order to accept or donate protons according to the classical Lowry-Bronsted principle of acid and base. At particular pH, one group remains predominantly ionized, but there exists a pH where the amino acids exist as largely neutral deuterionic form. This pH is known as isoelectric pH of the amino acids. Now coming to proteins and peptides. Peptides and proteins have become the drugs of choice for the treatment of numerous diseases as a result of their incredible selectivity and potency. We already discussed that the 20 different naturally occurring amino acids join with each other by peptide bond to form the protein or peptide. So generally the peptides which contains less than 20 amino acids, these are termed as peptides. However, the peptide chain which contains more than 50 amino acids chains sequence, they are termed as proteins. Proteins perform clinically important roles like internal protein network consisting of cytoskeleton maintains the cellular shape and physical integrity. Actin and myosin filaments form the contractile machinery of muscles, specifically of the heart muscle. Hemoglobin transport oxygen, while circulating antibodies rule out any foreign invaders. Enzymes, they catalyze several reactions that generates energy, synthesize and degrade biomolecules, etc. So, in short, we can say the proteins have both structural as well as dynamic functions. Different types of protein and peptides. So, based on the nature of the proteins, they are divided into either fibrous or globular protein, while peptides are classified according to the number of amino acids present, like oligo or polypeptides. So, oligopeptide means there are around 10 or less than 10 amino acids are there, otherwise they are polypeptides. Proteins acquire higher configuration for establishment of their integral functionality. These configurations are proceeded with hierarchy in the arrangement of the basic polypeptide chain into the secondary structure, further resulting into the tertiary structure. Tertiary structure with heteropolypeptide chain then forms the quaternary structure. This hierarchy of the protein structure is shown in this figure. Primary structure of the protein. The amino acid sequence written from N terminal on the left to the C terminal on the right is denoted as the primary structure of the protein. It is also known as the linear chain of the amino acid making up the protein sequence. Earlier, the amino acids were abbreviated using three-letter terminology. For example, ALA for the alanine and so on. But nowadays, a single letter abbreviation it is also used. For example, alanine it use only A, glycine it use G. So the denote the primary structure of the protein, the sequences can be written either ala, gly, phenyl, VL. So this is simply nothing but the peptide which contains alanine, glycine, phenylalanine and valine. Spatial arrangement of the polypeptide is referred as secondary structure of the protein. The free rotation of the bond angle allows the amino acids in the peptide backbone to attain the secondary structure. Secondary structure can be of two types, either alpha helix structure or beta pleated sheets. Alpha helix. A linear peptide chain either forms turns or coils leading to interaction of one amino acid with the fifth amino acid and so on in the sequence forming the alpha helical turn, or more commonly termed as alpha helix. Each carbonyl oxygen can hydrogen bond with NH hydrogen on the next turn of the coil. This figure shows four different models of the alpha helix showing different aspects of its structure. A. Formation of a right-handed alpha helix. The planes of the rigid peptide bonds are parallel to the long axis of the helix, depicted here as a vertical rod. B. Ball and stick model of a right-handed alpha helix showing the intra-chain hydrogen bonds. The repeat unit is a single turn of the helix, 3.6 residues. C. The alpha helix as viewed from one end. Looking down the longitudinal axis, note the position of R groups represented by purple spheres. This ball and stick model used to emphasize the helical arrangement gives the false impression that the helix is hollow because the balls do not represent the van der Waals radii of the individual atoms as the space filling model. D. Shows the atoms in the center of the alpha helix are in very close contact. Another type of secondary structure is beta pleated sheets. In this, the amide backbone of two or more polypeptide chains, they are associated by hydrogen bonds, are placed one above another like a sheet structure. So based on the loop or turn between on plane and another plane of the sheet that are either parallel or anti-parallel beta sheets. So it can have either anti-parallel or parallel arrangement. So in anti-parallel, N2C and C2N terminal, they are joined together, but in parallel arrangement, N2C and N2C, both chains are having N2C configuration and they are attached together. These proteins can also have the super secondary structure, which is a compact three-dimensional protein structure of several adjacent elements of a secondary structure. So these secondary structures are generally smaller than the protein domain or a subunit that is called the tertiary structure. The examples include beta hairpins, alpha helix hairpins, beta alpha beta motifs, or helix turn helix. Tertiary and quaternary structure of the protein. Tertiary structure refers to the entire three-dimensional conformation of a polypeptide. It indicates its three-dimensional space, how secondary structural features, alpha helix, beta pleated sheets, bands, turns, loops, coils, they assemble to form a domain and has these domains relate especially to one another. So tertiary proteins are generally either fibrous or globular. Fibrous proteins are insoluble and they are generally function as a structural component. However, these globular proteins, these are soluble, they are coiled into compact spherical shapes with hydrophobic groups oriented inwards and hydrophilic groups oriented outwards towards the aqueous environment of the cell. Quaternary structure, it is nothing but the joining of two or more polypeptide chains together with the help of either non-covalent aggregation or because of the disulfide bonds. This figure shows the different levels, various levels of the protein organization. Primary structure, as you can see, simple adjoining of the amino acids. Secondary structure, either alpha helix or pleated sheets. This is simply the coiling or folding into the primary structure. Then tertiary protein, which is having the different motifs or domains or pockets are there. And finally the quaternary structure, the two peptide chains or protein chains, they are joined together either by disulfide linkage or several non-covalent interactions. This figure also shows the similar thing, different levels of the protein architecture: primary structure, secondary structure, tertiary structure, and quaternary structure. So generally, disulfide bonds are involved in quaternary structure. Besides this, there are several non-covalent interaction like ionic interaction that leads to the salt bridges, or hydrophobic interaction, hydrogen bonding, van der Waals forces, aromatic interaction, or aromatic amino groups. So these are responsible for the protein structure and stability and leads to the formation of the quaternary structure of the protein. Now we will discuss recent scenario in the protein and peptides as therapeutics. Protein pharmaceuticals. According to the website of Drug Bank, www.drugbank.ca, there are around 150 FDA approved protein drugs are available in the market, and out of these more than 60% are the recombinant proteins. Protein pharmaceutical market is currently approaching around $130 billion per year, and by the end of 2015 it is expected to reach around 158 US billion dollar per year. Protein pharmaceuticals can be either vaccines, that includes peptides, part of the proteins or killed bacteria, or peptides like oxytocin, pitocin. They can be blood products like factor 10, factor 8, gamma globulin, serum albumin, etc. It can be different types of enzymes or recombinant therapeutic proteins like humulin, interferon, etc. Now we discuss the classification of the protein therapeutics. It can be classified in four major categories. Group 1, the protein therapeutics with enzymatic or regulatory activity. Group 2, these are the protein therapeutics with a special targeting activity. Group 3 constitutes the protein vaccine, and last the group 4, which are the protein diagnostic, proteins which are used in the diagnostic. Group 1, protein therapeutics with enzymatic or regulatory activity, they can be further subclassified as group 1A, 1B, 1C. So group 1A, these are the proteins which are used to replace the deficient or abnormal protein. So the example include the endocrine or metabolic disorder with a defined molecular etiologies dominated by group A. The classical example is insulin. Group 1B, that augments an existing pathway. So group 1B is dominated by the therapies that augment the hematological and endocrine pathway and immune responses. The example include interferon and growth factor therapies. Group 1C, these provide a novel functional activity. So these demonstrate the rational use of the naturally occurring proteins to modify the pathophysiology of the human disease. This table shows the examples of group 1A, that is the protein therapeutics which are used to replace a deficient or abnormal protein. So mostly the insulin, several growth hormones, these are the examples. In this table, the therapeutic or endocrine disorder along with the trade name, function and examples of the clinical use, these are summarized. This table summarizes the group 1B, the protein therapeutics which augments an existing pathway. So several examples like erythropoietin and so many other growth factors, these are given in this table. This table summarizes the group 1C, the protein therapeutics which provides a novel function or activity. So the examples include botulinum toxin, collagenase, hyaluronidase, papain, etc. Protein therapeutics with a special targeting activity are categorized in group 2, which are further subclassified in group 2A and group 2B. So group 2A interfere with the molecule or organism, and group 2B they deliver other compounds or protein. Example, the group 2A therapeutics use their special targeting activity to interfere with the molecules or organisms by binding specifically to them and blocking their function, targeting them for destruction or stimulating a signaling pathway. The best example is the monoclonal antibodies. However, group 2 therapeutics deliver the other compounds or protein to a specific site. Group 2A, this table summarizes the protein therapeutics that interfere with a molecule or organism. So several examples are given, and we can see mostly these are having application in the treatment of different types of cancer. This table summarizes the examples of group 2B, that are the protein therapeutics that deliver other compounds or proteins. So this mostly includes the monoclonal antibodies that denileukin, which is having the effect in the treatment of cancer. Similarly, tocilizumab monoclonal antibody, it also helps in the treatment of the different type of cancers. Group 3 protein therapeutics constitute the protein vaccine, which are further subclassified in group 3A, 3B and 3C. So group 3A, these are the conventional vaccine which are used to protect against a deleterious foreign agent. The classical example is hepatitis B vaccine or polio vaccine or any other vaccine you can name. Group 3B, this is used specifically to treat the autoimmune disorders, and group 3C, these are used for the cancer, means cancer vaccine. So the recombinant vaccines that provides a broad protection against the infectious agent, these are the group 3A vaccines, and the individualized vaccine against cancer are also available, and the proteins that can induce the protection against an overactive immune system that attacks its own body or self. It cannot distinguish these are the foreign invaders or this is the body own cells. So these are leads to the autoimmune disorders, and this helps in the understanding the molecular mechanism that allow the immune system to protect the body against the infectious disease and cancer. This table summarizes various protein vaccines. So on the above panel, you can see the examples of protein vaccines which are used for protection against the general infection like hepatitis B surface antigen, HPV vaccine, etc. On the lower panel, you can see the vaccines which are used for the treatment of autoimmune diseases like anti-rhesus immunoglobulin G. This is the main example. Group 4 category of the protein therapeutics includes the protein diagnostic. So these are the proteins which are not used to treat any disease, but these are the purified and recombinant proteins. These are used for the medical diagnosis, both in vivo as well as in vitro. So these are invaluable in decision making process that precedes for the treatment and management of many disease, because we can say the correct treatment can happen only after the correct diagnosis. Okay, so these are used for the diagnostic purpose, not for the any treatment purpose. So several stimulatory proteins hormones are used to diagnose the endocrine disorders. This table summarizes the group 4 protein pharmaceuticals that are basically the diagnostic agents. So several diagnostic agents can be utilized. For example, in vivo infectious disease diagnosis like recombinant purified protein derivatives. This had been utilized for the early diagnosis of the tuberculosis exposure. Various hormones like glucagon, growth hormone releasing hormone, secretin, they can be utilized. So glucagon has the application as a diagnostic aid to slow gastrointestinal motility in the radiographic studies and reversal of the hypoglycemia. Similarly, the growth hormone releasing hormone, it is helpful in the diagnosis of the defective growth hormone secretion. On the other hand, this thyroid stimulating hormone or thyrotropin, it is having the application as adjunctive diagnostic for the serum thyroglobulin testing in the follow-up patients with well-differentiated thyroid cancer. So students, let's summarize what we have learned out of this module. First thing, protein are the biomacromolecular polymeric chain with repetitive units of the amino acids which are linked via peptide bond. Amino acids, these are the basic units of the polypeptide, are named according to the location of the amino group like alpha, beta or gamma. The protein and peptide products have proven to be very successful and demonstrate the significant advantage due to their high potency. And according to the website of drug bank, that is www.drugbank.ca, there are approximately 150 FDA approved protein drugs are available in the market. The continuous advance in the biotechnology will produce more protein and peptide that would be difficult to administer by the conventional mean, and increased therapeutic potential they have been available.